Swayam Prabha. Digital India. Educated India.
Hello learners, today we are going to discuss about the products of evolution. This is the 7th module from the paper Evolutionary Biology. In the previous lectures, we have talked about what do we mean by evolution, what are the various evidences of evolution, how variation serve as raw material for evolutionary processes, what are the forces of evolution both in qualitative and the quantitative terms. Now let us move ahead to discuss what do we get out of evolution, or in other words, what is the product of evolution. I am Dr. Sudhir Verma, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. In the present lecture, we are going to learn about the patterns of evolutionary changes. Now, depending on the taxonomical hierarchy where these evolutionary changes are taking place, we call them as microevolution, macroevolution, or further the megaevolution. We will discuss all these three types. In microevolutionary changes, we will discuss about interpopulation variations, declines, ring species, etc. In macroevolution, or which is also called as adaptive radiation, we will take the example of Darwin's finches, and we will discuss about speciation as a process. We will also discuss about the tempo of speciation, various isolating mechanisms, etc. Before that, we will also discuss about the concepts of species and the subspecies, sibling species, etc. Also, we are going to discuss about the process of megaevolution. Evolution can be of two types, that is microevolution or macroevolution. A third type is also there which we call as megaevolution. These types are based on the level of taxonomical hierarchy where these evolutionary changes are taking place. If the evolutionary changes are occurring at the population level or the subspecies level, then we call them as microevolution. Whereas in case of macroevolution, the pattern of evolution occurs at or above the species level. So the evolutionary changes occurring at species or above species level are termed as macroevolutionary changes, whereas the evolutionary changes happening below species level or at the population level are microevolutionary changes. Let us talk about microevolution first. As we just mentioned, the microevolutionary processes are the evolutionary changes within the population. So there are two words that you need to focus upon. One is the evolutionary change and second is the population. Let us talk about these two to understand the phenomena of microevolution. Let's first talk about what do we mean by population. A population is a group of conspecific organisms occupying a well-defined geographical region and exhibiting reproductive continuity from generation to generation. Now I have used some words here like conspecific organisms, means the organisms or the population which are belonging to the same species. They are occupying a well-defined geographical region. That means they all are present in one geographical region. They do share that geographical area and they do exhibit the reproductive continuity. That means they can interbreed and this reproductive continuity goes from generation to generation. A population is usually made up of several demes. That means a population can be further divided into various demes or the groups. These demes or groups are partially isolated from each other, and this isolation is more of geographical isolation. So mostly a geographical barrier operates between two demes, and this geographical barrier limits the gene flow between these demes. But this barrier or this limitation of gene flow between demes is not sufficient enough which can result in speciation. That means these demes cannot be so separated that they evolve as different species. Though the barrier is there which inhibits the free gene flow, but it is still allowed to interbreed with each other. The demes are also called as basic unit of population. Just like we use cell as a basic unit of an organism, same ways deme is a basic unit of a population. So in totality, we can define demes as community of interbreeding individuals of a population or a species at a given locality sharing a common gene pool. So this definition combines all these characteristics of demes. That means they are community of interbreeding individuals. They are sharing the geographical regions. That means they are local in nature and they do share a common gene pool. Here is an example of frogs population in two different ponds. Pond A and Pond B are close by or nearby located ponds. Both ponds are inhabited by a frog population. Each frog population in each pond is a deme here. So they are interbreeding among themselves. So pond A individuals are interbreeding among themselves. Pond B individuals are breeding among themselves, and both of them are a deme. But these two ponds are isolated by a geographical barrier, but this barrier is not that strong enough so that they can evolve as two different species. Means the frogs can come out of this geographical barrier and they can breed with each other as well. So this barrier though existing, but it is not strong enough that it can lead to two different species in two different ponds here. There is another term which we use as clines. Regular and gradual stepwise modifications exhibited by a group of local population or demes from one part to another in their geographical range. Assume a population is there which is inhabiting a long geographical range, and the individuals show some kind of morphological differences when we move from one place to another one. So there are regular and gradual stepwise modifications which these members of demes represent when we move from one part to another in the geographical range. Or we can define clines as a geographical gradient in the frequency of a gene or in the average value of a character. So if the character changes across the geographical gradient, or the character which is being regulated by a particular gene, the frequency of that gene is changing across the geographical gradient, leads to clines. The term clines was coined by Julian Huxley in the year 1938. Clines can arise for different reasons. For example, the natural selection favors a slightly different form along the gradient. That means if the population are residing across a long geographical area, the local environment can favor a particular organism. So natural selection might be operating locally across the geographical gradient that is being discussed. Also, the cline can arise if two forms are adapted to different environments, separated in space, and because of gene flow or the migration. In simple words, you can understand that there are geographical isolations, or you can understand it by this, that the organisms are located across a long geographical area, and the moment you move from one area, local area, to another local area, you see some difference in the character or the frequency of genes in the organisms. So they are called as clines. It will be more clear with an example here. Here we are taking the example of yarrow plant, or which is also called as Achillea in botanical terms. This yarrow plant is found across a long geographical range, and the range which is inhabiting is the North American area. As you can see, the height of this yarrow plant, in the above diagram we are showing the mean height of yarrow plant in centimeters. So you can see there is a variety of height, and in the below diagram you see the altitude or the average height of the area where a particular yarrow plant species is present. So depending on which area it is inhabiting, the height of yarrow plant is getting altered. So this graded change in height of plant across this long geographical axis is representing an example of clines. Now here we are taking an example of height of a plant. It can be any character, or it can be change in the frequency of any gene across the geographical axis. Clines can also be further divided into continuous clines or the discontinuous clines. And even further, the continuous and discontinuous clines can be classified as horizontally stepped, obliquely stepped, and so on. So what do we mean by this continuous and discontinuous clines? The continuous clines, all populations of the species are able to interbreed, and there is a gene flow throughout the entire range of the species. Means if the organisms, or if the individuals or the populations of that species which are present across a long geographical area, all these populations are able to interbreed with each other, and gene flow is possible throughout the entire range. Then we call this as continuous clines. Whereas in case of discontinuous clines, the populations of species are allopatric. That means there is some barrier, geographical barrier existing between them, and hence there is little or no gene flow amongst the population. In other words, you can understand it like, if all the populations in clines are in continuous range both geographically and reproductively, then they result in continuous clines. Whereas on the other hand, if the populations are having some geographical isolations between them, which leads to reproductive barrier as well, which does not allow the continuous gene flow between them, then they are called as discontinuous clines. And further, as I just mentioned, they are classified as horizontal stepped clines, obliquely stepped clines, in both continuous as well as discontinuous clines. There is another term which we call as ring species, which is also called as Resenkreis, which is a German term for ring species only. A ring species is a connected series of neighboring populations, each of which interbreeds with closely sited related populations, but for which there exist at least two end populations in the series which are too distantly related to interbreed, though there is a potential gene flow between each linked population. Now try to understand it like this. Consider a circle, and on different points of periphery there are different populations. Each population can interbreed with the closely sighted populations. So on left hand side or onto the right hand side population, the one can breed. But there are two ant populations of these circle, and these two ant populations are so distantly related that they cannot interbreed with each other. Though there is a potential gene flow between each linked population, but these ant population are unable to breed with each other, and they lead to ring species. These non-breeding, though genetically connected ants populations may coexist in the same region. That means they are sympatric in nature, but they are closing a ring. Now let us try to understand it with some diagrams here. As you can see here in the circle in the middle diagram, let us see that in this Arctic region, there is a species which is called as Larus gulls, which you can see in the picture onto the right side. Let's say there are 7 different groups or populations of that species. Number 1, 2, 3, 4, 5, 6 and 7. And as you can see, these dots represent the area where they are inhabiting. Now, first species, that is say Larus fuscus, can interbreed with the second population, that is Larus fuscus but Siberian population. Second one can interbreed with first one and third one. Same ways, third one can breed with second one and fourth one and so on. But the species 1 and 7, which are present on two different ends of this circle, they are so distant, they are so unrelated to each other that they do not breed with each other. As you can see onto the image onto the right side, there is herring gull, which is Larus argentatus, which is onto the front side, and lesser black-hacked gull, which is Larus fuscus, which is present on the behind side. In Norway, these two phenotypes are having clear differences. They do represent two different ends of this ring. So they are called as ring species just because of the appearance of a ring or circular kind of appearance, which is representing the geographical range of the populations. Another example of ring species can be Encetina salamanders. As you can see in the image here, there are different populations of Encetina salamanders, and you can see the geographical area which is being represented by different colors here. Each population is inhabiting a particular geographical area here. Each one of them can breed with the neighboring populations. But the two ant population, again in the circular form, if you imagine, the two ant populations are so distantly related that they do not prefer to breed with each other. Thus, they also represent as an example of ring species here. Now, let us talk about the second component of our definition. Remember, we said microevolution are the evolutionary changes which are occurring at the level of population. And so far, we discussed what do we mean by population, what are the demes, what are the clines, what are the ring species, and so on. Now, let us discuss the second component of microevolution, which means the evolutionary change. So, what kind of change are we talking about? Microevolution is the change in allele frequencies that occurs over time within a population. So, all sorts of gene or allele frequencies that are changing within a population, they do come under microevolution, and this is the change which we are talking about at the level of population, which comprises of microevolution. This change happens over a relatively short amount of time compared to the changes which are termed as macroevolution. Now, this relatively short amount of time is in the evolutionary terms. It is not just like our clock or watch, which represents time in certain minutes or hours or seconds. Here we are talking about time in terms of evolution. So they are relatively short as compared to macroevolution, but otherwise they are large timings. This change in allele frequency is due to four different processes, and these four evolutionary processes are mutation, selection, which can be natural as well as artificial, it can be gene flow, that is the migration, and the genetic drift. In the previous lectures, we have discussed all these. What do we mean by mutations? What do we mean by natural and artificial selection? How do we define the phenomena of migration and genetic drift? Let us not go in the details of what do they mean, but we need to discuss how they change the allele frequency and what is the effect on the population. So let's talk about mutation first and how it changes the allele frequencies in the population. If the only evolutionary force on a population is mutation, the allele frequency changes with time. Eventually, it reaches equilibrium and is determined by forward and reverse mutation. Remember, there can be two types of mutation. That is the forward one, which converts the wild type to the mutant type, and there can be reverse mutation, which can revert back the effect from mutant to the wild form. So there is an equilibrium ultimately. The effect of typical mutation rates on Hardy-Weinberg equilibrium is negligible, and many generations are required for a population to reach mutational equilibrium. You must remember, we have already discussed what do we mean by Hardy-Weinberg equilibrium. So we are not going to discuss it again, but in terms of mutation, yes, they do alter this Hardy-Weinberg equilibrium. Second thing, which is migration. So migration also changes the allelic frequencies in the population. How? Because it introduces alleles from other population. If the migrants are coming to a particular population, obviously they are bringing their alleles or genes to that population. And when they breed with this population, they are going to have changes in the allele frequency of that population. The magnitude of change due to migration depends on both the extent of migration and the difference in the allelic frequencies between the source and the recipient population. So what was the initial difference between allelic frequency and how much the migration is happening? These two factors define the magnitude of change. Migration decreases genetic differences between the populations, because if two populations are migrating here and there, obviously they are decreasing the genetic differences, and it increases genetic variations within a population because new and new migrants are coming, so it increases the variations within the population. Moving ahead, the genetic drift, which is being represented by small population or the founder effect or the bottleneck effect, they also change the allele frequencies within a population. It decreases the genetic variation through fixation of alleles within a population. You must remember, one of the effect of genetic drift is it fixes the alleles in these populations. So since it is fixing one allele, it is going to decrease the genetic variations. On the other hand, it increases the genetic divergence between the populations. Moving ahead, the fourth evolutionary force is natural selection, and that also changes the allele frequency. The rate at which allele frequency changes due to selection depends upon the intensity of selection and the dominance relation between the genotypes. Here, the change in allele frequency for different type of dominance with constant selection coefficient is shown in this diagram here. As you can see, the dominant alleles increase rapidly through selection because both the homozygotes and the heterozygotes are being favored, as you can see here in the green line. The purple line shows alleles with incomplete dominance. With incomplete dominance, the alleles increase at a lower rate. Third line, which is represented here with the red color, is for the recessive alleles. Recessive alleles increase at the lowest rate because only the homozygote is being favored by the selection. So it depends on the intensity of selection and what is the dominance relation between the genotype, whether they are dominant or homozygotes or dominant or recessive homozygotes or the heterozygotes. So in this table, we can summarize the effects of evolutionary forces on allelic frequencies within the population, both in terms of short-term effect and the long-term effects. All these evolutionary forces, that is mutation, migration, genetic drift and natural selection, do have a short-term effect in terms of change in allelic frequencies. The long-term effect differs. For example, mutation leads to equilibrium between the forward and reverse mutations. Migration leads to an equilibrium when allelic frequencies of source and the recipient populations are equal. Genetic drift has a long-term effect in terms of fixation of one allele, and the natural selection differs in terms of its effect. In case of directional selection, it is leading to fixation of an allele. In case of over-dominant selection, it leads to equilibrium. In the next image, which is shown here, you can see the effect of evolutionary forces on genetic variation within the population and the genetic divergence between the populations. On the left side, as you can see, there is an increased genetic variation and decreased genetic variations. So that means we are talking about variations here in terms of increase or decrease. On the upper panel, you can see it is within the populations and between the populations. Mutation, migration, and some types of natural selection increase the genetic variation within the populations, whereas genetic drift and some types of natural selection decrease genetic variations within the populations. On the other hand, if you compare the population, then between the populations, mutation, genetic drift and some type of natural selection increase the genetic variations. Whereas the phenomena like migration, some types of natural selection, they do decrease the genetic variations between the populations. That means they decrease the divergence. Now let us move ahead to the next pattern of evolutionary change, which we call as macroevolution. Macroevolution is the evolutionary change at and above the species level. You all must have read about species, and if I ask you to define species, you might have a number of definitions to define the species. In the image here onto the right side, this is the hierarchy of classification, which goes from species to genus to family, order, class, phylum, kingdom, domain, and the overall life form. So, species, if you see, is the basic unit of classification. You must have done all these classification and taxonomy part in your previous classes, and when you classify your organism, you generally go to species level, and that is why we call the species as the basic unit of classification and a taxonomic rank of an organism, as well as a unit of biodiversity. For example, if I use the name Canis familiaris, which is the name for domestic dog, this is a species. But the problem is how to define a species. This is fine that this is a basic unit of classification. This is the taxonomic rank of organism. It is a unit of biodiversity. These all are correct. But how to define that this is the definition of a species? When these characters are met or when these parameters are met, we call that as a species. How to define that? For that, different biologists have given different criteria. Jerry Coyne and Alan Orr in the year 2004 noted that species can be defined in a way that enables us to classify organisms systematically. So it provides us a systematic way of classification of organisms. Corresponds to discrete group of similar organisms. That means we can define the similar organisms as a discrete group or a particular species. It helps us to understand how discrete clusters of organisms arise in nature. It represents products of evolutionary history and it applies to the largest possible variety of organisms. Remember, it is highly unlikely that any one species concept will serve most of these purposes. We do have n number of species concept, almost 30 different concepts are there to define a species, a modern concept we are talking about. But it is very unlikely that any one species concept will serve most of these purposes which we have narrated here. Now, let us talk about various species concepts that have been put forward to define a particular species.